Okay, so this video correlates to the third presentation um, regarding chapter 2 in part 1 of the book. Um, so if we take a look at the genes, we'll see that they have two components. One is called an exon and one is called an intron. And those components, both components, have more than one uh, um, sequence reoccurring through the gene. When we examine the RNA molecules, they do not have the introns. So the introns are taken out before. The introns are those who represent the 5' and the 3' ends of the molecule. All right. So we talked about taking out sequences, cutting out, and we call that splicing, right? So the introns are removed by splicing when we're producing the mature RNA molecule. And some say that they act as a safety mechanism, adding another step before the final RNA is produced, giving the system more time to think about what it's doing and what exactly it needs and what it doesn't need. So both mRNA and RNA are very similar, just that the mRNA is the RNA without the introns, right? Once we splice out the introns, the ends of the exons are reconnected, right? So on slide number 9, you are referred to a movie. I hope you enjoy it. And on slide number 10, you can see that we're saying that the gene is actually the exons plus the introns. The exons are coding matter, protein coding, right? And the introns are non-coding matter. So transcription advances from the 5' end to the 3' end of the mRNA, and sometimes even beyond the 3' end. Any cleavage of the RNA beyond the 3' or polyadenylation are important to gene expression. So even though the 3' end kind of symbolizes the end of the coding area, there is importance to processes that are utilized and done on the sequence, made on the sequence, to the sequence after the 3', and they can change matters of gene expression. Right, so we are on slide 12, and we're saying that since DNA has introns, it means that the distances between sequences, especially protein coding sequences in DNA, do not correspond directly to what we're going to see in the protein, right? Because when we get to the protein phase, and even before, in between, we have sequences, introns, that are being spliced out. So the DNA has a lot of sequences that do not code for protein that are taken out, and then when you compare that sequence in DNA and the protein, you can't find a high correspondence between the two. Of course, that the protein is the product of the gene, but the gene has to be so-called processed, right? Go through transcription and then translation of the mRNA in order to produce the right protein. So the precursor RNA represents the length of the gene and not mRNA. Why? Because we just said mRNA is the RNA without the introns. So if we want a representative of the gene's length, we either have to see DNA, right? That includes everything, if it doesn't have mutations, if it's perfect, and RNA. But mRNA is closer to representing the protein rather than the gene. So splicing is very accurate and specific for every RNA molecule. It's not as if splicing occurs at the same point no matter which RNA molecule we're talking about. So since we define the exons as the sequences that have the protein coding elements, then we're talking about mutations in the exons that will actually affect the protein, right? But mutations in the introns may affect the production of mRNA, okay? And the splicing of exons together. Why? Because they might cause shifts in the reading frame, right? And that might include the introns. And if the splicing point is wrong, anything can go wrong, theoretically. Of course, when we have intron mutations, they can affect only the allele that carries it, that carries that intron. Okay? So the majority of mutations are single base substitutions. You already know what that means, right? And the connections, or what we call junctions, of introns and exons are affected. So these kind of mutations are usually deleterious, meaning that they will lead to a delete mutation, meaning that some kind of sequence will be taken out, erased, so to speak, excluded from the sequence. And you know that too. So when we look at all the mutations that are known to man that cause diseases, 50% of those affect the splicing process. So that's quite a lot, all right? It's quite a lot. All right, so right now we are on slide number 14. In a lot of cases, it is the exon that is deleted and the intron is included, or a stop codon is inserted, and we already spoke about the effects of stop codon, right? So the strand can be broken down and cleaved to certain short sequences of known length. These are called fragments. Okay, so we have ways of recognizing these fragments and cleaving or breaking down the strand into them. Okay, remember we talked about recognition sites and specific enzymes, so basically we're talking about the same thing. We can recognize certain sequences that have certain roles in the whole molecule, and we can use the specific enzymes that operate on those specific sites to create those fragments. This will aid creating the physical map, or to physically map DNA, and the enzymes that actually do it are called restriction endonucleases. Okay, so the restriction endonucleases are those who know how to produce those fragments that we're talking about. Okay, so we are on slide 15. These enzymes are sequence specific and will cut the strand wherever the same sequence occurs. So everywhere that they identify a certain sequence, they're going to cut it from the two ends and take it out. And these sequences, you can ask, okay, why do that? Because if we need to repair something or we need it for recombination, right? Or reverse recombination, those can be used to produce something else. So it's kind of looking at spare parts in a way. So once a restriction map can be produced for a certain molecule, we can understand the distances between the different factors and components of the gene. And that allows us to control a lot of lab techniques needed for different different roles or goals, amongst others, also research. Okay, so the distances are measured in base pairs. Okay, so we write BP for short. But what happens if we have, distances that are calculated or counted in thousands of base pairs, meaning 10 in the third degree? So we can use a unit that's called a kilobase, KB, which is 10 in the third degree BP. All right. Don't be confused with blood pressure. Not the same. All right. What if we have millions of base pairs in a certain sequence? So we'll just use the unit of megabase, which is 10 in the sixth degree. All right. So that's kind of easy. All right. We also find sequences with hundreds of base pairs or less than hundreds. So let's say we have a container right now with all kinds of fragments in it, and we want to identify them. We want to know their length, or we know the length and we want to know if they're there, right? If they exist in the samples that we have. So a technique used quite a lot is called gel electrophoresis, and it's used to separate the fragments. And the idea is that you create a gel, and it has loading docks in it, usually 8. It depends on how much you want to create, but basically usually it has about 8 docks or 10. And you insert your samples in there, and this gel sits in liquid and is connected to an electrical current. One is a plus and one is a minus. And when that current is applied to the gel, it makes the fragments move. But exactly like any other thing that any object that you'd like to move, the bigger the object, theoretically, I'll be accurate. If all the objects that you're trying to move are made from the same material, right? The bigger the object, the harder it's going to be to move it. So the same thing goes for the fragments. The smaller fragments are going to go faster and farther in the gel, and the bigger ones are going to be closer. All right. So the bigger the fragment, the harder it is to make it move, and it will advance slower. So you're advised on slide 17 to see a movie, enjoy it, and then you have the picture here. So let's go over it, and I'll start from we're talking about slide 18 right now, okay? So let's start at the upper left corner. So we see in this case 4 loading wells or sample wells, which is the origin of where they're going to start moving. So you take a pipette and you load the samples there. Okay, so for instance, that can be DNA samples, right? And you can see that the current is going from the negative electrode, which is where we load the samples, and it's going to go to the positive one that is on the far end of the gel. So the movement direction is from minus to plus. Okay, and we can see that the heaviest fragments are closest to the minus, and the lightest fragments are as far as possible from the minus. They're closer to the plus. Okay, and by this technique we can actually profile a sample. Okay, so this map that we're creating may include mutation fragments, if at all. It might and it might not, right? But it does not indicate about the function or dysfunction, meaning that even if you have a perfect map of a gene, it means that you know the fragments. You don't know if it's functional or dysfunctional yet, right? Because you don't know the outcome. It's just a map of a gene. Okay. So when we're talking about an uninterrupted gene, we're talking about a gene that corresponds perfectly or exactly with the mRNA. Okay. So such a gene will have what? Little to no introns, because we don't want it interrupted. Okay, remember we're talking about the gene that is uninterrupted. Okay, the majority of genes by far are interrupted genes. Okay, the length of genes between species, even if theoretically the exons are exactly the same, still vary in size and length. Why? Because the introns vary in size and number for every strand, for every gene, and for every genome. So the major factor making a difference between the length and sizes of genes are the introns. When we're comparing genes that have similar structure and function, right? The interrupted genes are present at almost all levels of DNA and RNA, and even mitochondrial DNA. So we reached the end of slide 20. We'll stop here and continue in the next video.